For our morning session, and that is observational research in immunopsychiatry, and then we're talking about human observational research. The next two talks are prepared by Michael Benros and myself, and we will give you an overview of the state of the art. What do we know from mechanistic, from observational research in the field of immunopsychiatry? What do we not know yet, and what could we learn in future research activities? So I would like to invite Michael to share slides. Thank you. Michael, if you can put on your camera also, then maybe we can see you. And I can introduce you. You are a professor at the Copenhagen University Hospital, MD and PhD in Denmark. And I hope I don't see you yet, so maybe
you can see me. I think
yeah, you need to move in and say something,
okay, yeah.
And the screen pops up. So welcome, and yeah, I know you will be starting, so the floor is yours.
Yeah, thank you very much. Yes, and Brenda and I will have the next presentation. And first, I'll start off with briefly summarizing the current state of the art in clinical studies and observational studies. And then Brenda will be asking the questions, and then we will discuss it afterwards. So first, starting off from epidemiology, where there's strong evidence of associations between different immune exposures and subsequent increased risk of psychiatric disorders. And these two graphs are shown in the nationwide Danish studies, where we use the whole Danish population as one large cohort. And in the first slide on the left, we look at psychotic disorders as an outcome. And here you can see in the last blue bar, if a person has autoimmune disease and 3 or more hospital contacts for infections, the subsequent risk of a psychotic disorder diagnosis is increased by 3.5 times. But as you can also see on the red bars, each number of infections by themselves increases the risk in a dose-response relationship. And these results are, of course, adjusted for social factors and a parental history of mental disorders, etc. I mean, you look at the right graph, here we have the outcome of mood disorders. Here you also see that the risk increases in a dose-response associations with the number of infections. And here we actually only looked at the number of infections, but if instead look at, you have to have different types of infections, different contacts, then if you have 5 or more different hospital contacts with different types of infections, then your risk of subsequent mood disorders is increased by 5 times. And here in this subsequent study, we looked at all mental disorders, where we actually see a similar pattern. But here we also show the temporal associations. So you can see the first 3 months after a hospital for contact for infections, you actually have a 5.6 times increased risk of a new onset mental disorders. And if it's only prescriptions, so you're actually not hospitalized, then the increased risk is increased by 3.7 times. And of course, here the reference group is a bit different. And also here you see that the risk increases in a dose-response relationships across the mental disorders. I mean, looking at infections in the brain, CNS infections, we show that CNS infections increases the risk of a new onset mental disorders by 40%. And then when wondering whether the risk is highest if the infection is inside the brain, encephalitis, or if it's in the meninges, then the risk is mostly increased if you have an encephalitis, so infection in the brain. And also in these prior studies, it's actually viral CNS infections that were associated with the highest risk of mental disorders. And this was actually in contrast to what we see in the periphery, where it's bacterial infections that contribute to the highest risk for subsequent mental disorders. And in the periphery, it might be that bacterial infections induces the highest inflammatory load, so immune components are then more likely to actually reach the brain and pass a more leaky blood-brain barrier. But as you can also see here in this graph, with CNS infections, there's a very, very strong temporal association. So the first half year after you have had a CNS infection, there's a 27 times increased risk of getting a new onset mental disorders. And this risk actually remains significant up to 20 years after the CNS infections, but of course much stronger in the beginning. And then when looking at the genetics, we have used a Danish cohort, which is representative of the whole Danish population, so not a selected cohort, as you see in the PGC samples of 65,000 individuals. And here we show that the SNP heritability of infections are only 4%. So that means just getting infections is, of course, mostly environmental, where you transmit the disease from an individual to another. So with the genetics contained in a GWAS, this only explains 4%. But for this small genetic contribution, there's actually a moderate genetic correlations between the genes that are associated with susceptibility for infections and the genes that are associated with mental disorders. So it seems to play a small part at least. And here when looking at the specific HLA region, so in our sample, when doing adjustment for multiple testing, we only find significant associations with autism and intellectual disability. But if we instead look at this HLA region and only look at the nominal significant HLA SNPs, then there are multiple associations with schizophrenia and ADHD. But as we can also see here on this graph, when we only do the GWAS on our sample on mental disorders, we see no specific associations, not a very strong association with the HLA region. But if we instead restrict it to people with mental disorders and autoimmune diseases, then we see a very strong association in the HLA region. So it's probably subgroups of individuals that have chronic immune conditions that are genetically related, and this subgroup can possibly be broadened out. And then we're looking at the studies on the blood. Here we looked at all blood tests that were conducted when you were admitted first time to a hospital with bipolar disorders. Then there was an average CRP of 3.5. And for schizophrenia, there was an average CRP of 3.1. And for depression, it was 2.8. Whereas healthy individuals would normally have CRP below 1. And these point estimates are a bit higher than what you see in clinical studies, since in this studies we included all patients and when at the point when they were acutely admitted. So the state of the disease might also impact these immune markers. But also importantly, we see that the CRP levels are associated with mortality. So if you have a CRP levels between 3 to 10, you have an increased mortality of 56%. And if you have a CRP level of above 10 when you're first time admitted in psychiatry, your subsequent mortality rate is increased by 2 times. So it's also something we can use already now clinically. So we actually need to investigate these individuals with high CRP to detect the underlying somatic condition to improve the current treatments. And also when looking in blood, here's a meta-analysis by another group by Wittenberg, where they have looked at the transcriptomics data. And here they show that there's associations between the innate immune system and depression, and also the neutrophil related gene networks. But it's also important to remember that they did it as a meta-analysis, but not as you would regularly do on genetic data, where you have included all data points. So here they only included the published data points in the studies, as you can see here. So all these studies do not have measured the same markers. So subsequent studies need to conducts similar studies on raw genetic data to get a more precise picture of the associations. But still, it goes very well and in line with what you see on the normal immune markers in blood of people with mental disorders. And also, this is a meta-analysis by Miller, where they look at the state of trait. So this is the graph for psychosis, I believe. So the red bars, if you have with the blood test, are conducted during acute relapse, and the blue one are during a first episode. And here you see that many of these cytokines are increased during these acute phases. But if you look at the green ones, it's post-treatment, then these cytokine levels are actually not very different or even decreased. So you can see, and these immune markers are very fluctuant and vary throughout the day. So the state of the mental illness clearly matters for this. And here, when looking at CSF studies investigating the cerebrospinal fluid, we did a systematic review and meta-analysis of all studies conducted. But many of the studies had investigated different markers, so it's only some of them where we could actually reliably look at. But what goes true between both schizophrenia and mood disorders is that the signs of increased permeability of the blood-brain barrier on a group level, measured by the albumin ratio between the CSF and blood. And also for schizophrenia, as the example here, there was a significant increased level of IL-6 and IL-8. And only 2 of the CSF studies actually looked at how many were re-diagnosed after a CSF test. And there, between 3 and 6% actually received a new diagnosis, which was an immunological diagnosis. So it can have clinical importance, at least in a smaller group. And here we did a CSF register-based study on 15,000 individuals and looked at the CSF results. And this is, of course, taken by clinical indication, so it has potential bias in it. And there we see for mental disorders, there's an increased risk of subsequent mental disorders after a abnormal CSF test, but the risk is also increased after one within the normal levels. So you can see the CSF markers that we use on the routine is not indicative of psychiatric disorders, but are mainly used to actually detect underlying CNS infections. And also there were a little bit more specific with neurological diseases. So we need to investigate for biomarkers that are actually specifically associated with mental disorders, whereas the ones we have now are actually more used for differential diagnostic purposes. And then for the microbiome, Nikolova et al. Performed a meta-analysis of 34 studies showing a small decrease in microbial richness and a small decrease in phylogenetic diversity, and also signs of depleted levels of Faecalibacterium and Corbococcus, which were across many of the mental disorders. But for the microbiome studies, it's important to remember that longitudinal studies are lacking, and there were none where they actually had microbiome measures before the onset of the mental disorders. So much of this can also be influenced by diet and behavior. And we're looking at neuroimaging studies related for immunopsychiatry. The slide on the left were one of the first that came out, where the picture fitted very well, with the schizophrenia having increased levels of microglia activation, and the high-risk group was just in between healthy and schizophrenia. But subsequent studies with meta-analysis for psychosis actually indicate that there's no difference on the TSPO levels, at least detected significantly across these studies. Whereas for depression, there's signs of increased TSPO levels, which are markers of microglia activation in 6 out of 7 studies. So currently the evidence is largest for the influence of microglia in depression based on these scanning studies. And then we're looking at post-mortem brain studies. There are limited evidence for microglial activation and lymphocytic infiltration across all the mental disorders, so it's not something that all the patients have, but there are signs of upregulation of pro-inflammatory markers and also upregulation of gene expressions. And studies indicate that there is a subgroup of the patients that have particularly high immune signatures in the brain. So it's very likely these subgroups we need to identify. And then this is just shortly where we have performed the two largest meta-analyses on RCTs of anti-inflammatory treatment. And this is for depression and depressive symptoms covering almost 10,000 patients, whereas the 600 that had clinical depression, there was actually a standard mean difference effect of minus 0.64, which is a moderate to large effect. And for the depressive symptoms, it was 0.41, which is a moderate effect. So there seemed to be some solid evidence that this is the way to look further on these RCTs. But for psychotic disorders, the evidence is a bit stronger, where we show significant associations, particularly for neurosteroids and NAC. And here we use mean difference and PANSS, so it's not the standard mean difference. But what I think is very interesting, when we do meta-regression of the size of the study for psychotic disorders, we show that the larger the study is, the smaller were the effects. And there were many very small studies. So we need more larger studies and also studies that measure immune markers. So what is lacking? Detailed immune characterization in the same individual from brain to the periphery, both including CSF, blood, and microbiome, with longitudinal assessment and cross-disorder samples. And we need to stratify to identify these immune-related subgroups based on relatively stable immune markers and or clinical symptoms. And RCTs based on immune-related biomarkers, where there are several ongoing now. So we're looking much forward to these results. And also to have an improved and more standardized methodology in sample collection and analysis, which is currently much needed in the field. And of course, most importantly, we need to identify the exact molecular causal mechanisms, so we can actually better design drugs for this. But there are some good biomarkers that we can also already use now. So if you have brain-reactive antibodies in the CSF, such as anti-NMDA, or if you have a CSF cell count above 5 or other marked CSF pathology, or if CSF infections are detected, or if there are ongoing peripheral infections noticed by a high CRP or by clinical indications, or autoimmune antibodies indicative of undetected autoimmune diseases, or indications shown at the MRI or EEG, then further evaluation and very likely treatment is indicated. But this would likely be in a smaller group of the patients. So the current question is if we can broaden out this group of patients that can have a different treatment than today. And one of the examples to actually try to identify this is our cyclame studies, where we look across tissues from CSF to blood to microbiome, and across diagnosis from psychotic to affective disorders, and both have neurological examinations and digital and massive biobanking, which would then also eventually go into a big machine learning framework to identify immune-related subgroups, and also hopefully giving us mechanistic insights. Oh, yeah. And now Brenda will continue the talk.
Yes, thank you. Thank you, Michael, for giving, I think, a great overview of what we know about observational evidence linking immune system to psychiatry. And I think there obviously are quite some questions that remain, and that observational research can at least play an important role in as well. I'm not saying that observational research can do it all. We need experiments and we need mechanistic research as well. But I do think it's good to talk about what we could, in addition, learn from observational studies. And I think one of the questions, obviously, is what biological associations mean. I think lots of what Michael has shown were mainly cross-sectional associations between biomarkers of the immune system with mental conditions. And obviously, we need to be aware of the fact that a biological association can mean different things. It can indicate that a biological marker is an etiological risk factor. Biological marker can be a scar of psychiatric disease, but it can also be an innocent bystander. So in order to really look a little bit further at the causality aspects, I think it's very good to keep in mind that also from observational research, we can do quite a few things to tap into a more causal inference evidence for the immunopsychiatry links. And that could be done by really utilizing some key features of observational research. And I will point out a few. For instance, think about the time trends, that temporality that we can test with observational data. Lots of what we know right now, and what also has been summarized by Michael, is cross-sectional. And I think we do have more and more longitudinal cohorts that we can utilize to really look better at the causal ordering over time. We can also look at consistency more. And I think we can also utilize molecular epidemiology to better understand and examine the molecular mechanisms behind the associations using cohort data. So one of the next questions that we could look at, and I think that's on the next slide, is, as I mentioned, the ordering of associations over time. There, I think there's quite clear evidence that biomarkers such as CRP and several interleukins are linked to, for instance, depression, as you have heard from earlier evidence. But there's not that much longitudinal data that really tells us, is it the inflammatory marker that is predicting also subsequent course and development of depression over time? And also, the question, can interleukin 6, for instance, or another biomarker of inflammation, predict chronicity of depression over time? So how does this ordering look like? And I'm just showing an example of findings that we have that looked at the ordering of associations over time using the NESTA data, which is an observational patient control cohort that we have in the Netherlands, where we have several waves of data collection, as you can see on the upper part of the slide, 9 years of follow-up, where we have looked at multiple waves of data and addressed the question, is depression predicting interleukin 6 levels 2 years later, and is interleukin 6 predicting depression chronicity 2 years later? This is work done by Femke Lamers. I think she's also here in the Zoom link. Together with Femke Lamers and Yuri Milaneschi, we have examined quite in detail some of these remaining questions using NESTA data. And in this study, we found both evidence that depression is predicting higher levels of IL-6 2 years later, but also IL-6 is predicting an increased risk of subsequent 2 years chronicity. So the ordering is really bidirectional. I think it's important to utilize longitudinal data to examine the temporal ordering over time more. Another question, and that's the next slide, is really the consistency of associations across different markers. There are lots of observational cohort studies where people have either looked at CRP or at IL-6 or tumor necrosis factor alpha, which I think are among the most commonly assessed biomarkers, at least in the larger cohorts. And that is partly because those are accessible. They can be measured using high sensitivity assays. And they seem to be they seem to be rather valid measures to measure. However, it's sometimes very difficult to really look at consistency of findings across different cohorts because all the cohorts are using different biomarkers. And I think we also need to consider whether it is making sense to look at cumulative inflammation markers, where we consider evidence of having high levels of 2 or 3 or more biomarkers to be more pronounced evidence of inflammation. Because very likely, if CRP and interleukin-6 and maybe some other interleukins or tumor necrosis factor indicators are all high, I think the evidence of a more systemic chronic inflammation is even higher. So really checking on consistency across different markers and think about consistency across markers, I think is another next step that we need to build on more. And that is also an illustration from the NESTA study published last year in Journal of Affective Disorders, where Christian Vinkers created a cumulative inflammation index that is summarizing the number of inflammatory markers that are upregulated. And that actually showed the strongest association with total depression severity score and also with atypical energy-related severity dimension within our NESTA cohort. The third question that we could also and should also have a look at is that, like Michael already indicated, there is evidence of immune system dysfunction across multiple mental disorders. However, lots of studies are zooming in on only one psychiatric condition, and that makes really the comparison and the question, is the immune system involvement similar or not across different mental conditions, still very difficult to address. And I think this is also a question that we could approach in cohort studies when we, for instance, would measure similar markers and hopefully even using the same lab and using biobank samples, for instance, and apply that to different psychiatric patients. Then we can learn better than we currently do whether the immune system involvement is similar or not, and how certain biomarkers compare in depression, in bipolar disorder, in schizophrenia, and in autism disorder. And that is simply a question that we can't address very clearly yet. I think this is illustrated in this meta-analysis where we looked at 2 mental conditions, schizophrenia and depression, and we compared the evidence of involvement of immune biomarkers across these 2 conditions in drug-naive patients, so that we ruled out the impact that antipsychotic and antidepressant treatment could have on immune system biomarkers. And as you can see from this slide, well, first of all, in some of these studies, there were really limited numbers of patients. But also, if you then compare findings, it sometimes is true that biomarkers are showing the same effect size and the same direction of association in schizophrenia and depression. However, there was also some evidence for certain cytokines to show actually a contrasting association in depression and schizophrenia. So, for instance, the findings for IL-4, the findings for TGF, and maybe some findings for some of the other biomarkers are not completely consistent across schizophrenia and depression. And that can still simply indicate that we don't have enough uniform evidence and strong enough tested data where we really do comparable approaches to measure biomarkers across different patient groups. But I do think that this is still a question that we need to address, because we need to be certain to what extent immune system involvement is similar or not across different disorders. So that's also a question for further observation research. Then the fourth question is really this causality question again. And I think the cohort studies that we have available do provide the opportunity to use genetics as a tool to examine causality better. And this is coming from the field of Mendelian randomization. I think the genetics has the advantage that we clearly know where the causality started. So the genes are fixed at conception. There is no reverse causation. And by using genetics as a tool, we can actually explore causality better. So how that works is that when you measure specific genetic markers, such as the IL-6 receptor gene, you know that this is a tool, again, where no reverse causation is possible. And we can then test whether IL-6 receptor gene is linked to IL-6, but also whether the IL-6 receptor gene is linked to outcome depression. But then we know certainly that the causality really is indicating that already from the genetic basis, there is a link with an outcome like depression. And this is something that we can do now more and more, because there are a few very large cohort studies available where we have genetic genome-wide data on, and also within the context of the genetics consortium, the PGC, where we have very large cohorts now where we have detailed genetic information, we have inflammation information, and we have depression information. And I think we can use genetics more to test causality, and also test to what symptom dimensions our genetic tools point us to. And that is actually evidenced here by a recent paper that Yuri Milaneschi led using data from Nesta and the UK Biobank, where he tested, in this case, the IL-6 receptor gene, and found associations with involvement with suicidal ideation, with sleeping problems, and with fatigue as the symptoms of depression to be strongly linked to IL-6 receptor gene variation. And I think this is another innovative way of using our cohort data to further disentangle causality. Next question is really the underlying mechanisms by utilizing better large-scale omics. And I think, again, there are quite some large-scale cohorts now where there is detailed epigenetics, transcriptomics, proteomics, metabolomics, microbiomics, and also information on the exosome, where we can further investigate using those omics and molecular epidemiology tools to better test what is underlying the immune dysregulation in mental conditions. And I think the next slide shows some examples of studies where I think we could have access to within our consortium, because I know that the ECMP immunopsychiatry working group has actually made an inventory of the available cohorts that we have within our network and the types of data we have within these cohorts. And we actually do have more and more information on some of these underlying omics levels. And I think it's actually very useful to look at that, and for future research, apply a common approach where we collaborate and standardize our assessment, where we move beyond one marker and where we look beyond diagnostic criteria and really explore dimensions of symptoms, and do that together so that we really can learn from these large-scale omics opportunities and apply them to our immunopsychiatry questions. And then last, the question, and that is actually not the least important. In fact, I think it's one of the main questions we really have to address, and for which we could use observational data more, is really the question, who are they? And I think, as was clear already from Michael's talk, is that we know that not all the depressed or schizophrenia patients or bipolar patients, not all of them are immune dysregulated. We need to find out what is the subgroup of patients for whom immune dysregulation is most prominent. And there are some nice publications out there, and I did see in the chat already a question on what is the prevalence of the inflammation in a general population. Well, we know that if you zoom in on depressed people, like has been done by Itaruli in the UK Biobank and in a meta-analysis by Gholam Kandakar's group, that probably in depressed patients, it's not more than probably a quarter of all the depressed patients who have levels of, or evidence, if you look at the CRP level, evidence of some systemic inflammation. So we need to find out who that quarter of patients are. And it's also good to realize that 75% does not fulfill these criteria. So this comes back also to what Peter has mentioned, that it's really not obviously a necessity to have immune dysregulation to suffer from a psychiatric condition. So better separating and pointing towards who are those people with immune inflammatory dysregulation, I think is really a topic that is very important for next research questions, and also to allow us to isolate those who may best benefit from potential intervention. And in our group, and that's last slide on next slide. In our group, we have looked at that question also, and by really zooming in on symptomatology, correlates, pathophysiology, and genetics. And there we have identified also about a quarter to 1/3 of all the depressed persons with more higher risk of immune metabolic dysregulation, with evidence of leptin and insulin resistance. And they seem to suffer more from the neurovegetative symptoms, such as increased appetite and weight, low energy, hypersomnia, and leader paralysis. They are also the ones who have much more metabolic syndrome also over time. And they are the ones with the highest risk of genetic risk of obesity and metabolic dysregulation. So by diving into our patient groups and finding out how do these people with immune inflammatory dysregulations look like, what are their symptoms, what is their genetic profile, their pathophysiology profile precisely, I think we can learn more about who they are. And this is just an example from our research, but I think there are many more symptom dimensions to explore that are relevant and that are in the literature indicated to be potentially more strongly related to immune inflammatory dysregulation. So if you can press the button, Michael. Yeah. So I think this is really a question where we need to zoom in on specific symptom dimensions such as anhedonia, melancholia, and panic symptoms, because there are quite some reports on a potential more relevant role of some of these symptom profiles for immune inflammatory dysregulation, so that we can also better understand what it is in the symptom phenotype that is more strongly linked to immune inflammatory dysregulation. So if we summarize that on the last slide, and this is partly repetitive of what Michael already mentioned, is what is needed from the observational perspective of research is that we really indeed synchronize and standardize some of our longitudinal studies and apply multiple measures of immune markers, that we also have a methodological state-of-the-art in collecting samples so that we better synchronize with what we are doing and not everybody is doing their own thing, making results across studies not comparable, that we really move beyond a single marker and look at composite scores so that we can better identify underlying mechanisms of inflammation, and that we use, for instance, the multi-omics opportunities to better dissect molecular causal mechanisms, and that very importantly, we really identify the group of persons to whom immune inflammatory dysregulation is truly present, and really know whom to target in case we want to do experimental approaches. So with that, I would like to stop. I think that's the last slide.
Yes, I'll just stop sharing. Yeah.
And Michael and I are both back. I obviously have not been able to look at the chat while I was talking. But I do think some general questions may come back at the end. Michael, I'm not sure whether you already looked at the chat and identified one or two questions we should.
No, I can just look at it now. Start.
I know that there are some people mentioning that they agree with Janet's comments. I think Janet probably is on the Zoom call. So maybe Janet, you can mention the point you raised.
Yeah, it's been a point I've been raising more and more loudly the past time. I think the problem with the whole field is that we are trying to get the biology to fit the psychiatric diagnosis, and when it doesn't fit, we throw away the biology. And we're doing this again and again and again and again. And when we find associations, we're making the mistake of extrapolating it to the entire group. And that recently I saw a meta-analysis where they do show, that there is increased inflammatory cytokines. And then they conclude that this shows that depression is an inflammatory disorder. And that is so not true. Because yes, there is a group that have inflammatory markers, but there's a huge group, and actually the large group does not. And as the first talker mentioned, there's a group that also has a low inflammatory marker profiles, and that's probably also contributing. So I think this inflammation 0-1 way of thinking of the whole problem is a huge problem as well, because that you have the innate immune system, you have the adaptive immune system, they're all working in balance against each other. And IL-8 and IL-6 can go entirely opposite directions in certain patient groups. And I really think we have to be really careful about the way we're thinking about this, and not be in too much of a hurry to create new groups. I think we have to stop and stratify and allow that kind of messiness where we deconstruct the whole thing and then rebuild it up and be willing to sort of let go of our old groups. I recently got a reviewer comment, which was because I use a transdiagnostic approach, and they insisted that if I had used psychosis patients alone, the group would have been much more biologically homogeneous. And this is like a lie we have told.
Just going back to the unidimension or unidiagnostic approach again. Yeah.
Yeah, it's we've convinced ourselves that the diagnosis system we have is biologically correct. And if you think that, you haven't sat with the data, because the data will tell you the exact opposite. This is not biology homogeneous groups we've created. We have to let these go. So that was my
I do see a lot of recognition, and I think this really points towards questions on, we need to do much better cross disorder approaches, but also explore within diagnosis groups who are the ones who show most dysregulation and how to identify those.
Yeah.
I think, Hemo, you have comments based on that as well. I think I see your raised hand.
Yes, can you hear me?
Yes.
Yeah, I guess so. Yeah, I totally agree actually with Janet. When I see the enormous, huge cohorts people want to study just simply with cytokines, I think this is very insufficient. I think the work in mood inflame and mood stratification and TRIC-8, focusing particularly on the cellular immune system, on monocytes and T cells and aging T cells and Th1 cells and Th17 cells, which haven't been at all discussed here, clearly shows that in some of the what you call anti-inflammatory or non-immune depression, you have many signs of immune dysregulation, but just another way of dysregulation. The same holds true for the interferon. You have interferon-driven inflammatory responses, but you also have pyroptosis-driven inflammatory responses, and we can all recognize them in different groups of patients. We at least identify now 4 endoimmunotypes in depression, 3 in schizophrenia, and only in each one overlaps with the other, you see? So exactly what Janet says, it's too complicated. Well, we have to not to think of any psychiatric diagnosis. No, I start to believe that you have to think of immune endocrine diagnoses. And you can subdivide it. But of course, I see it's very complicated. The one approach is many more patients and one simple CRP measurement, and then we do it all. Or we do also in smaller groups a much more broader approach. And of course, to harmonize cellular immune parameters is very difficult, but it's doable.
Yeah. Well, it's very likely we need both approaches, and they need to feed on each other. So I think that's ultimately probably the best way of combining, the deep phenotyping approach with also raising the question, what are the patients that it applies to then? And I think we really need both approaches combined in both best worlds. I would propose that, Michael, you wanted to...
Yeah, I just wanted to say that I very much agree, and I think that was actually the essence of our talk. So that we need, but this was the current evidence, and now we need larger studies with much more detailed measurements and also symptom levels, so we can actually go across the diagnosis and identify immune-related groups.
Yep.
I'd like to just jump in and ask the question though. Do you think these large studies should continue to select on symptoms, or is it not time, because there's a huge amount of money to run these studies? Is it not time to start zooming in a bit so that we can conduct studies that have a chance of coming out as positive?
Yeah. I mean, I gave the example with the cyclane studies we are looking at, where we look at across the whole affective spectrum and the whole psychotic spectrum and have specific symptoms. So I think it's actually studies like that where you can go across the diagnosis and also look at specific symptoms that are related to specific immune markers that might difference between groups. But for that, you need to do it convincingly, you need to have very large sample size with statistics. So, I mean, it will also be needed to combine studies, because even if you have a sample size of 1,000 with these very detailed measurements, it is still likely not large enough, but that's a start at least.
Yeah, and I don't think it's only sample size, it's also uniform assessments. I think we are currently not measuring the same things in schizophrenia patients as in depression patients. We're not measuring the same dimensional scales, for instance, which really makes it difficult to test whether the symptom involvement and the symptom relevance is across disorders is really the same as linked with immunoinflammatory dysregulation. And that, I think, is also something that needs to change if we really want to make this cross-disorder approach. I guess we are right now already in the discussion part of this morning session. I would propose that we take a 10-minute break and then we continue with the combined talk by Bernard Baun and Carmine Variante on intervention research. And then we have at the end of the morning still a general 30-minute discussion on immunopsychiatry. So for now, please refresh. There is a bio break. You can get a cup of coffee. Unfortunately, I can't offer it to you. I would have loved to do that in Amsterdam, but we're not ready yet with COVID. But everybody, I'll see you back at 10:50. So let's take a 9-minute break, and then we continue with the next. Thank you.